welcome to our channel. Today we are starting Neisseria. As we have already covered the previous lots of bacteria, and today's topic is Neisseria. Okay, after completing today's class, you will get a great overview regarding the Neisseria. Just subscribe our channel for daily classes. Okay, okay. We are coming to understanding the Neisseria. Firstly, see here, that is a genus. Whenever we are understanding any kinds of bacteria, we need to understand about the basic introduction of the particular bacteria. And I am again saying you that I basically takes the class in whole English only for you all guys, and the English is completely understandable in English, so that every kinds of student or professionals can attend the particular classes. That is my only motto to taking the class in English. Okay, so that please subscribe as well as please see until or unless it's complete. See, first one is that it's a gram negative bacteria. Okay, Neisseria is a gram negative bacteria. How it look like? That is look like a diplococci. Diplo defining that there should be two coccus, so that we are saying that the diplococci, kidney bean or coffee bean shape. It's basically looking like. Okay, it's a very important things we need to understand. After that, it's basically founded into the human and animal mucosal surface. Getting my point? Now we are coming. There is a two type of Neisseria. First one is the pathogenic, and second one is the commensal. First one is the pathogenic, means they are causing the disease, so that we are saying that the pathogenic. And second one is the commensal, it defining that they basically stray, they basically take nutrition, they basically whole thing they are performing in any kinds of host body, but they are not causing any kinds of disease. That is called commensal. Okay, so that we are discussing about the pathogenic Neisseria, because if they are not causing any kinds of disease, how do we as well as why we will study the particular organism? They are good, right? We should not study of the particular organism. See, first one is the Neisseria gonorrhoeae. They causes the gonorrhea. Okay, second one is the Neisseria meningitidis. They basically causes the meningitis and spectemia. Understood? After that, now we are coming to the commensal organism. They basically not causing any kind of disease. Okay, like Neisseria lactamica, Neisseria sicca. Okay, that's all are the things. After that, I am coming to the morphology section. As I have already told you that Neisseria basically gram negative bacteria. Got it? That is a pink or red in the gram stain, so that it's called gram negative bacteria. Secondly, the arrangement should be diplococci. Diplococci means, see here, that is one coccus, that is one coccus. Whenever two coccus are attached, that is called diplococci. Okay, pear shape. After that, they are non-motile, means they should not do any kinds of motility. Okay, after that, spore absent. Getting my point? After that, capsule in the Neisseria meningitidis, they are encapsulated, means they have the capsule. Neisseria gonorrhoeae, they are non-capsulated, means they are have not capsule. Getting my point? Because we are discussing in only these two Neisseria. One is Neisseria meningitidis, another one is a Neisseria gonorrhoeae. Now we are coming to the size, that is a 0.6 to 1 micrometer size. Okay, that's all are the morphology related to the Neisseria. Now I am coming to the cultural characteristics. As whenever we are discussing any kinds of bacteriology, we basically discuss in the particular form, right? We have to understand firstly morphology, we have already understood. Now we are coming to the cultural characteristics. See, it is a fastidious. Fastidious means they cannot grow in the ordinary media. They require the enriched media. Getting my point? See, they are aerobic or microaerophilic, means they require oxygen or little level of oxygen they require. Okay, and 5 to 10% CO2 enhance the growth, means capnophilic. Whenever we are adding 5 to 10% of the carbon dioxide, then the bacterial growth become increases. What are the temperatures, sir? See here, 35 to 37 degree temperature. Okay, what are the colony morphology for the Neisseria gonorrhoeae? You will find out that small means very much small. Translate means you will find out from the one side to another side. After that, see here, grayish you will find out. Okay, and in the Neisseria meningitis, how much as well as which kind of culture characteristics you will find out. See, larger. After that opaque and sometimes mucoid. Sometimes you will find out the mucoid colony as well. Okay, you have to just see that is a colony, that is the cultural characteristics. Now I am coming which kinds of media as well as which kinds of agar basically uses for the Nigeria. First one, see here, the chocolate agar we are using supports the both species, that is species for the Neisseria gonorrhoeae and Neisseria meningitis both. Okay, after that, Martin agar, that is selective for the Neisseria gonorrhoeae. Please keep in mind that because they have some antibiotic which inhibit the another bacteria. Got it? After that, that is the blood agar we have to use this, because see, Neisseria meningitis basically grow into the blood agar, and Neisseria gonorrhoeae are basically growing lessly, means poorly. Getting my point? That's all are the medium we have to use this. Okay, chocolate agar we have understood, Martin agar we have understood, and blood agar we have already understood. Now we are coming to the biochemical reaction. See, we have already understood the morphology, we have already understood the cultural characteristics. Now it's the high time to discuss about the biochemical reaction. Okay, now see, first one is the oxidase, they basically positive oxidase. Catalase you have, that is positive. And this kinds of biochemical test I have already discussed in my previous lecture. Just look down upon the particular lecture, and that is available in the YouTube as well. You have to just look down upon the biochemical test, you will find out every kinds of biochemical tests are present there. Okay, after that, superoxole test, that is also positive. Okay, after the DNA test, getting my point, DNA test, that basically positive for the Neisseria gonorrhoeae. Getting my point, that basically positive for the Neisseria gonorrhoeae. Okay, see, so that oxidase test is positive, we have understood. Catalase test is also positive, we have understood. After that, superosol test is also positive, but DNA test is negative. Okay, DNA test is negative, please keep in mind that. After that, now I am coming to the carbohydrate fermentation. See, glucose only fermented, that is Neisseria gonorrhoeae, and glucose and maltose basically ferment the Neisseria meningitis, and glucose, maltose and lactose fermentation basically happening by the Neisseria lactamica. Okay, that's all are the biochemical reaction. Now we are coming to the disease caused by the one is Neisseria meningitis we have to understand, and another one is the Neisseria gonorrhoeae we have to understand. That is widely used as well as widely stated disease. You might listen lots of time into your surroundings, right? Neisseria meningitis, yeah, you they basically causes the meningitis. Okay, second one is the meningococcoemia. Why we are saying that meningococcoemia? Meninges, they basically the inflammation are happening into the meninges. Where the meninges is present? The meninges is present into the brain layer. Getting my point? And the inflammation are happening into the meninges, so that it's called the meningitis. Meningococcoemia defining that into the meninges layer there should be increasing the diplococci, so that it's called meningococcoemia. After that, Waterhouse Fridson syndrome, it is also one type of syndrome. They basically hemorrhage are happening into the adrenal gland. Got it? After the pneumonia might be also occurred, it's too much rare due to the Neisseria meningitis. Okay, that's all are the Neisseria meningitis, or we can say that the meningitis. You might find out that that is the diplococci basically founded into the particular meninges layer of the brain. Getting my point? So that it's called Neisseria meningitis. Now we are coming to the disease state by the Neisseria gonorrhoeae. Okay, they basically causes the gonorrhea. They basically causes gonorrhea. Please keep in mind that they are sexually transmitted infection. Please keep in mind that. Into the men, you will find out the erythritis. Okay, and the past discharge basically happen. Painful urination will be happen. Okay, whenever you will excrete out the urine, you will become feel the pain. Okay, in the women, cervicitis, PID, infertility, these kinds of disease basically, or these kinds of symptom might be occurred by the gonorrhea disease or by the Neisseria gonorrhea. Okay, after that, disseminated gonococcal infection means whenever they are spreaded throughout the whole body, that is called disseminated gonococcal infection. Okay, bacteremia will occur. Bacteria will find out into the arthas, that is arthritis, rashes. After that, tenosynovitis, these kinds of symptoms will be occurred. Getting my point? After the neonatal conjunctivitis should be also occurred. Neonatal conjunctivitis, you will find out. See here, neonatal conjunctivitis. Okay, ophthalmia of the neonatum acquired during the birth. Whenever the birth are happening, you will find out the neonatal conjunctivitis due to the Neisseria gonorrhea. May cause the blindness if untreated. If the these kinds of neonatal conjunctivitis are untreated or ophthalmia is untreated, then they might also causes the blindness as well. Getting my point? That's all are regarding the Neisseria gonorrhea, as well as that's all are the things related to the gonorrhea disease. Now we are coming to the lab diagnosis. Firstly, we have to collect the sample for the Neisseria gonorrhea. We have to collect the urethral, cervical, rectal, throat, and conjunctival swab. Where the infections are occurring, we have to collect the swab from the particular place. Okay, for the Nigerian meningitis, you have to collect the CSF blood or nasopharyngeal swab. Okay, where the infection are occurring, you have to collect the sample from there. We will find out that gram negative diplococci inside the neutrophil. Getting my point? After the culture, we have to do the chocolate agar as well as Thayer Martin media. I have already told you that they are capnophilic. Whenever we are adding the 5 to 10% CO2, then we will find out that more good result. Okay, biochemical test, I have already told you that oxidase positive and sugar fermentation pattern, that is also positive. After that, into the molecular biological test, we have to do the NET, that is nucleic acid as amplification test. We may do. We can also do the PCR for the Neisseria meningitis. Okay, last one is the antigen detection. We may also do the antigen detection by the latex agglutination test. That's all are the thing related to the Neisseria meningitis as well as Neisseria gonorrhea. That's all are the lab diagnosis. Now we are already ending stage of the particular lecture. My main motto is to taking the overview of the particular bacteria. As we have already covered the lots of bacteria, just look down upon the channel, you will find out the another bacteria, and subscribe our channel, you will find out for more interesting lecture. Okay, see, first one is that we may give the vaccine for the Nigerian meningitis. There is a vaccine available, we may do. Okay, first one is the main ACWY against the serotype ACWY. Okay, these kinds of serotype basically present for the Nigerian meningitis, and we have to give the vaccination. After that, main B against the serotype B. Okay, please keep in mind that. After the safe sexual practice, we may do uses condom, as well as we have to limit our sexual partner for preventing the Nigeria. Getting my point? That's all are the thing for today's lecture. Thank you so much for attending the class. Subscribe our channel and just check the description box. Join our free group. You will get this note as well, and every kinds of vacancy update you may find out. Thank you so much for attending.